Well, good morning, Phoebe. Good morning, Larry.
Hello.
Good morning, Zach. Welcome, everybody. Jalen, good morning. So we get everybody up on the screen. Jordan Cole, Adelaide, Nava, and Jenna, good morning. Trying to get all this stuff going here. Let's see, a bunch of people in all at once here. Let's see, who did I miss? Olivia, good morning. Shane, good morning. Kathleen and Anna, Bradley, Margie. Spencer's gone from friends, just himself. I like that. Welcome, everybody. Glad you're here. There. Present. All right. We got just about everybody. Nope. One more. Well, I think we would be remiss not to address the enormous elephant that is sitting or was sitting, will continue to sit in the Capitol yesterday. And I don't know how best to address this, so I'm really just going to ask you a question as our check-in for today. Do you want to take some time to share your thoughts, your experience, your feelings around what happened in our nation's capital yesterday? Go ahead and drag that line to the spot that best represents your wishes and desires this morning. If you're just joining us, I know a couple people just popped in there a minute ago or a second ago. We're trying to address the big question, right? Do you want to spend just a few minutes here as we get started this morning, sharing, addressing your thoughts, your feelings about what happened in our nation's capital yesterday? Just kind of drag your line to the spot on that continuum that best represents your desires this morning. Kind of waiting for the lines to settle down a bit. Looks like they tend to be concentrated in the middle so far. So the majority doesn't want to share, but you want to hear what others think. That means somebody's going to have to share in order for us to hear what others think, right? I guess there's one person who's willing to share right now. Well, given, I had no idea how you were going to answer this question. So I don't know that this is the appropriate tool, but let's try it, since you want to know what other people are thinking, but you yourself may not want to share. There's this link to a current events Jamboard. It's really just a blank board at this point. I'm hoping you can click on it directly. If you can't, I can put you into student mode real quick. And maybe, I honestly don't know, maybe this is the best way to see what other people are thinking, but to share your thinking in a really safe place because it's really anonymous. So I would invite you, if you are interested, you can draw something, you can write something, you can post a sticky note. I bet you can even find an image quickly that you could add to the board that best summarizes your thinking or your feeling about what happened. Let me do this. If you can't, click on the link here. I'll drop the link into the chat too. See if that helps things a little bit. Try that. Again, this is not required. I had no idea how you were going to answer this question, but if you do want to see what other people are thinking or feeling, and you're willing to at least put something up there for other people to see, keeping in mind that it's anonymous, I would welcome you to take just these first couple minutes of class and try that out. Is that working a little bit better, I hope? See, a couple people are joining. Take people out of student mode here, just for the moment. We're not going to belabor this. If you're not interested in putting something up there, that's okay. There's something. Here, you can put it up so everybody can see it too. Gonna do a new share here. Let's go big so we can see it. Yeah, if you post something, it will superimpose it over the existing node. So just drag your node over. Thank you to those who have already shared. Again, you can draw something, you could put an image up, you could write something, post a sticky note. Ah, there it is. Thank you for resizing it. I couldn't see the rest of the text. I think Tommy Lauren, if I'm pronouncing that correctly, she's a political pundit.
Something like that. I just found this quote really ironic. She tweeted this out.
Okay, but don't these people have like jobs they have to go to?
Fair question. What's one way to take a personal day, I guess? Just take another minute here. We will do some biology today, I promise. I do appreciate that at least one person has shared that, you know, he, she, they couldn't do their homework, which I totally get. If you want to say something out loud, of course you can. Okay. Try to find a spot where you can post your final thought or your final contribution here. Thank you to those who have shared already. See if I can pull your faces up, at least those of you who have your faces on the screen. I don't really have any words specifically to share with you this morning. I was talking to another science teacher. Believe it or not, there aren't that many adults in the building, so I don't see a lot of people during the day. But I had a chance to briefly talk to another science teacher this morning, and I really posed a question because it's, I think it's one of my deepest concerns for you folks is, you know, most of you are 15, 16, 17 years old, right? And the last 5 years have been, you know, the formative years of your adolescence. This is what you've lived, you've experienced, you've seen, you've heard. And I'm 42, you know, I'm an old fogey. And the only thing I really can share with you this morning is that what you saw yesterday, what you've seen for the last 5 years, it's not normal. It's not my lived experience. And that doesn't make it good or bad necessarily, but it's very different. I think it's historical in context. And I think it provides an opportunity for all of us, even though most of you can't vote quite yet, but you will be soon, to really reflect and consider how we want to move forward as a community, as a state, as a country. Because it didn't used to be this way, at least not in my lifetime. So that's really all I have. It seems like an opportunity for choice. Would anybody like to share anything else that they didn't get a chance to share already before we move forward?
I just wanted to kind of say something important. I think this is one of the first times in history that the votes or validity of the votes have been brought to question on the Senate floor like they had before. But I think in 2016, it was a peaceful transfer of power, and then the votes were brought into question. But this is the first time that the Senate floor was divided, and that's why it took so long, because this is usually one of the quickest things to pass through the Senate. And while they were debating, this happened. So I thought that was kind of interesting.
Anybody else? I'm going to switch over to our slides here again. Okay. Well, I'm hoping other teachers today will give you some time, some space to share your thinking. If not, let this be the spot where you did have a chance today. And I'm hoping that you found it at least a little bit useful. So thank you. All right. Well, we do have some biology for the day. I do hope you had a chance to watch the Edpuzzle video on cell membrane structure and function, because we want to use the content in that video and practice with it a bit today. Again, where the pattern that we developed last semester is the pattern that we're going to follow this semester. The Edpuzzle videos are the lesson. I strongly encourage you to watch them before you come to class, and so that we can work with that material, and you can ask questions about the material if you have it. I don't want to give you a big problem set to do for homework. That doesn't make any sense to me. I'd much rather we do the problems during the time that we have together, so that if you do have questions, you can ask. So what we want to focus on today is really see if I can pull up what we left with last time. This idea of, come on, buddy, life as being cellular. So as you think back to the videos that you had a chance to watch last time or finish watching after class last time, you know, the Miller-Urey experiment and experiments like them revealed that the conditions of the early Earth were totally appropriate for the very biological molecules that are required for life to come into existence. And the idea is that once those molecules existed, they could take the form of a membrane or take the form of a genetic material like RNA, and that genetic material actually had the ability to self-replicate. And as we watch this video on the screen right now, what I was trying to share with you is this notion of, well, once you have a self-replicating genetic material enclosed within a membrane, you can actually very simply visualize in real time under a microscope. I can get it to pause at just the right moment, and of course I didn't, so I'll just play it again. You can actually see those structures reproduce, make more of themselves. I was trying to pause it at just the right moment, because what you can actually even begin to see is that as that one structure becomes several, they're not all the same. That variation is introduced into the population through this reproductive event. And what we're looking at on this screen is the gradual, but relatively fast, accumulation or really growth, elongation of a membrane that surrounds a really simple cellular-like structure. So I'm going to see if I can pause it at the right moment this time so we can see that reproductive event. 1, 2, 3. I'm trying to get it. There, that's not bad. Right, so you can see 3 different structures have emerged from the one. So that's really what today is all about, is to dig a little bit deeper into what is that membrane that's enclosing the materials within, whether it's the earliest cell to exist on Earth or one of the cells in your body right now. What does that membrane look like? How does it work? And why is it important in the first place? So that's where we're gonna be going today. All right. So there's this term in biology called homeostasis. Often it's confused with equilibrium, and it shouldn't be. because the word balance is used to describe homeostasis. And that's really the role of the membrane as an outer boundary for our cells, whether it's, again, the cells in our body or the first cell to exist on Earth. It's an outer boundary. It separates the inside of the cell from the outside of the cell. So sometimes I refer to this as like a door person, right? It's regulating, it's controlling, it's deciding what gets in, but also what gets out, okay? So with that in mind, the first question I wanted to pose to you is that if you were to think about a really simple cell, I mean, literally I have a membrane with something on the inside, kind of a black circle on the inside. Maybe that's the RNA that's serving as a genetic material, okay? What sorts of molecules do you think need to be able to get into this cell? And what sorts of molecules do you think need to be able to get out of the cell? Because if the membrane is regulating what gets in and out, we have to begin to think about it and maybe just brainstorm here at the beginning of class, what sorts of molecules need to be able to get in and get out. So you can type, you can draw, you can use arrows to show the direction that you think these molecules are traveling. Take a few minutes here and brainstorm some molecules that you think have to either get in or out of a typical cell. And don't get stuck on specific molecular formulas or even specific names necessarily of molecules. It can be general here. The membrane is a boundary between the inside and outside of the cell. It's keeping some stuff in and it's keeping some stuff out. So what do you think needs to get in? What do you think needs to get out? Let's take a look at people have come up with so far. Yeah, food, right? That makes sense. Things that provide nutrition have to be able to get in. Waste has to be able to get out. Yeah, food, waste, water. Yeah, oxygen is a molecule. O2, one of your diatomics, right? Your Brinkelhoffs. I've been told that you had to memorize those in honors chemistry. And carbon dioxide has to get out. Now that's true for a typical typical animal cell, but for a plant cell, we can often actually think about it as almost being reversed, right? That oxygen is going to be coming out and CO2 is going to be coming in. Now in reality, plant cells are doing both, but certainly those molecules have to be able to move into and out of the cell. Yeah, glucose, cholesterol, I like that. Good thinking. Food, waste, nutrients. Water, yeah, water's a nutrient for sure. Ooh, I like that. Amino acids have to be able to get in. Got to build proteins after all. Got to have the building blocks present. Water, food, very nice. Water, I like how water has to be able to move into and out of the cell. You have arrows pointing in both directions. Very nice. There's that O2 coming in, O2 coming out. So maybe that's an example of a plant cell. And it's okay not to be sure. Water coming in. Yeah, good. So good thinking. Thank you for sharing. And this is not meant to be any sort of exhaustive list. We're going to encounter different molecules along the way that have to be able to get in or out of the cell. But ultimately, what we want to begin to think about today is how the cell membrane actually accomplishes that regulatory process, right? How is it determining what gets in and what gets out of the cell? Alrighty. So in a perfect world, we would be all together in the classroom and we would have some time to actually model not only what the cell membrane looks like, but what it is composed of. But given where we are right now, that is just not a possibility. So what I want to do instead is show you a short video clip. It's only 3 minutes long that really summarizes what I would have hoped you would have taken away from an in-class modeling exercise. Some of what you're about to see is going to be redundant. It was introduced in the Edpuzzle video that you've already watched, but it's a really short video clip, and I wanted to make sure that especially given what happened yesterday, if you didn't have a chance to watch the video, that we're all kind of sitting in the same place with the same information before we move forward. So we've done this before. I'm going to go ahead and press play. And if you can see and hear everything on your end, just give me a quick thumbs up. And again, this is a short video clip that summarizes the most important pieces of information in respect to the structure of the cell membrane. All right, here we go.
Welcome to Construction of the Cell Membrane. In this video, you'll learn the makeup and the basics of cell membrane function by examining the molecular structure of the cell membrane. It's a critical step to learning about the fundamental unit of life, the cell. The membrane's main job is to protect the cell from its surroundings. It separates the cell's interior from the outside environment and helps it keep its shape. The membrane is selectively permeable, which means it allows certain organic molecules to pass into and out of the interior. The cell membrane is mainly composed of lipids and proteins. Its framework consists of a double layer of phospholipids. Lipids make up 50% of most membranes, although depending on the location, they may make up to 80%. The remainder is made from proteins. Lipids make the membrane flexible, and the proteins help transfer molecules across the membrane. There are two major types of proteins: tightly coiled, rod-shaped fibrous proteins, which help give the cell support and shape, and the more compact, globular-shaped integral proteins and peripheral proteins. These help cells communicate and transport molecules. Examples include glycoprotein, pore protein, and channel protein. Because the cell membrane is mostly lipid, it only allows lipid-soluble substances like oxygen, carbon dioxide, and steroids to pass through. Water-soluble substances such as glucose, amino acids, ions, and water need the help of the various proteins in order to move across the membrane. Two layers of phospholipid molecules self-assemble so that their water-soluble or hydrophilic heads form the surface and interior of the membrane, and the water-insoluble or hydrophobic tails face each other. The fibrous proteins may span the entire membrane and serve as receptors for the cell. They communicate with the external environment using hormones and neurotransmitters. One type of globular protein forms pores to allow lipid-insoluble water molecules to pass through. Other integral proteins serve as channels and selectively support ions, including nutrients such as sugars or amino acids. Channel proteins are usually quite specific, so each one specializes in transporting a single substance or group of similar chemical substances. Globular proteins are peripheral, meaning they're associated with the surface of the cell. These include enzymes or glycoproteins. These proteins have carbohydrate associations that identify the cell. Cholesterol molecules are embedded in animal cell membranes, but not in plant cell membranes. They're found dispersed through the cell membrane and help strengthen the membrane wall. In addition, they, along with phospholipids, help make the membrane impermeable to water-soluble substances. Cell membranes are integral to cell health. With their combination of lipids, proteins, and cholesterol, they are able to complete a complex variety of tasks essential to life. Cell membranes keep the outside out and the inside in. You've completed construction of the cell membrane.
All right, so is that bringing up any questions for you, whether that was from the Edpuzzle video or just what you saw there a moment ago? Anything about the structure of the membrane that you're unsure about or curious about? Okay, so like I said a moment ago, in a perfect world, we would be building models of these cell membranes. And I've got little styrofoam balls and pipe cleaners and sticks and all these different things that we can use to actually construct a model of the membrane. But given where we are right now, that wasn't possible. So I wanted to share that video with you because I thought it did a nice job summarizing not just the overall structure of the membrane, but some of the different parts, some of the different molecules that are found within the membrane as well. But as you saw, the most important molecule that composes the membrane is the phospholipid. And the phospholipid is a molecule that actually resembles a fat. It resembles a lipid from last semester. It's a little bit different in that I kind of like this image over here on the right-hand side compared to the image on the left-hand side, because I think simple is better in this case. But really all it is a polar head and two non-polar tails. And this is important to us because as you saw, those molecules are going to arrange themselves into a bilayer. So two layers of these phospholipid molecules will form on their own, spontaneously. Nothing is building this structure, right? It forms all on its own. And that's the connection to what we did the other day, is that once these molecules are present, even on an early Earth, they were self-assembling into these structures, into the membrane in this case. So nothing has to happen in order for the structure to form. It forms all on its own. But as we think about these molecules coming together and giving rise to a membrane, what's important to note is which side of the phospholipid, since there are two layers of these phospholipid molecules, are facing the outside and the inside of the cell respectively, and then what these molecules are going to be interacting with as a consequence. So this is the first practice question that I wanted to share with you. So take a moment, study the diagram. I hope you can see the numbers on your screen. What do you think best represents the numbered areas in this diagram? So we think about the structure of the phospholipid and how these molecules come together to make a membrane. And if you have clarifying questions, don't hesitate to ask. So I can even draw. So this is one phospholipid, and this would be the other phospholipid that makes the bilayer, if that helps at all. All these little guys. Maybe you recognize that from chemistry. Those are water molecules, bent molecules. About half of people have responded. Try to get your answer in. All right. Let's see how you're doing here. All right, kind of a mixed bag. So before we all start changing your answers, which is what you're apt to do, right? Let's go back and take a look at the diagram. So the idea here is that if you have these phospholipid molecules, one part of the molecule, really the head of the molecule, so in this case, it's kind of the two circles that are together, that part of the molecule is polar or hydrophilic. Those two terms can be used interchangeably. And polar hydrophilic parts of molecules, or molecules in general, are going to be able to interact with water molecules because water is also a polar molecule, right? There's an unequal distribution of electrons across the structure of a water molecule. So what they're trying to show in this diagram is that in area 1, pull my pen up here again, in area 1, you've got the head of these phospholipid molecules interacting with water molecules. So polar water, polar hydrophilic head of the phospholipid. Maybe even in chemistry, you're familiar with the term like dissolves like. So polar dissolves polar. That's what's illustrated in this diagram here, that polar molecules interact with the polar parts of the phospholipid. The same thing, of course, is happening down here. So we don't know exactly where we are in terms of the cell membrane, but if this was the outside of the cell, then this might be the inside of the cell. And what that's telling us is that on either half of this bilayer, right, one layer of phospholipids is interacting with a watery environment on the outside of the cell. And on the inside of the cell, the inner layer of phospholipids on the inner layer of the bilayer is interacting with a watery environment on the inside of the cell. But what's super important is that when you look at area 2, sorry, I'm a little far there. Area 2, there's no water there, right? So the tails of the phospholipid molecules are non-polar. They're hydrophobic. They want nothing to do with water. So as we begin to make some connections to where we just left off, the cell membrane is able to serve as a barrier or a boundary between the internal and external environments of wherever that cell is located, because the outermost portion of this bilayer is polar, while the inner layer of the membrane is non-polar. So if something's going to cross the membrane, it has to be non-polar in respect to its chemical properties. There's really not any other way for the molecule to get across the membrane. So molecules like oxygen and carbon dioxide, they're non-polar. They're going to be able to cross the membrane. But molecules like amino acids, glucose, charged ions, they're really not going to be able to cross the membrane on their own, but they can cross the membrane if they get a little bit of help from one of those proteins that you saw illustrated in the video. So if we go back to the responses, now that people have changed their answers, 1 and 3 are hydrophilic. They're polar because they're interacting. Ah, okay. You all changed your answer, but you changed your answer due to peer pressure, not because it was the correct answer. So areas 1 and 3 are indeed hydrophilic. They're polar, right? That part of the phospholipid is interacting with water, but they're not forming covalent bonds with water. That's not what's happening. So area 2, that's the middle, that's not polar. So really it's gotta be answer A. 1 and 3 are polar, they're hydrophilic because those membrane molecules are aligned with the water molecules. They're not interacting covalently, they're simply interacting, it's not shown in this answer choice, but through hydrogen bonding. Okay, so resist peer pressure, especially if you answered A. And does this bring up any questions for you before we move forward? So the membrane is regulating what gets into and out of the cell based upon the chemical properties of the molecules that make up the membrane, the phospholipids. Part of the phospholipid is polar, go back a little bit, or hydrophilic, that part faces water on both the inside and outside of the cell, while the tails of these phospholipids are hydrophobic. They're non-polar. They're buried on the inside of the membrane, and thus they're serving a chemical barrier that determines ultimately what gets into and out of the cell. All right. Do we need to take a short break? Would that be helpful to stand up and stretch for a minute? Give me a thumbs up if it would be helpful. All right. So I've got 9:37. Why don't we take a quick break, stand up, stretch, get a drink of water, go to the restroom, and we'll come back together at 9:40. Alrighty, that was a short break, so let's come on back together. All right, so again, the cell membrane is a boundary between the inside and outside of the cell. The composition of the membrane actually helps determine what's going to get into and out of the cell, because we have these phospholipid molecules that form a spontaneous bilayer with the polar part of the molecule facing out and in, the non-polar part of the molecule making up the inside of the membrane. And thus what we end up with is a structure which determines what gets into and out of the cell based upon the chemical properties of what's trying to get into or out of the cell. So as we kind of expand outward, so far we've just kind of just focused on two-dimensional images of the membrane. I like this image on the screen right now because it gives you a chance to see that this bilayer is completely covering the surface of the cell, right, three-dimensionally. This is a spherical cell, and not all cells are spherical in structure, but you can begin to appreciate the fact that even in this cutaway view, which I think looks like a heart, I don't know, doesn't it look kind of like a heart to you too? That this phospholipid bilayer is covering the entire surface of the cell. Okay, all right. So another question to think about. Here is a two-dimensional view of the membrane again. Just checking and making sure people are getting this. How are these phospholipid molecules oriented relative to one another within the model? It's going to be a quick one. I'll give you 30 seconds. All right, your screen's going to lock because the timer went off. Let's see how we're doing. Okay, kind of a mixed bag again. So can't be A, because the non-polar portions of the phospholipid molecules, right, they're going to be buried on the inside of the membrane. So they have nothing to do with the internal and external environments that the cell finds itself in. The B makes sense, right? The hydrophilic phosphate groups, like the head part of the molecule, is attracted to the aqueous internal and external environment. Remember, aqueous from chemistry just means watery. So B makes sense. Embedded proteins attract the fatty acid tails of the phospholipids so the tails point away from the internal and external. That's not impossible. Certainly there might be parts of the protein, hydrophobic, non-polar parts of the protein that are interacting with the non-polar tails of the fatty acids. That doesn't really get at why the phospholipids themselves are arranged into the configuration that they are. B is a much better answer. And D can't be correct because the fatty acid tails are not hydrophilic, they are hydrophobic, they are non-polar, they want nothing to do with water. All right. So as we begin to build on this understanding, what we can begin to think about is that these molecules that make up the cell membrane, the phospholipids, because of their own intrinsic chemical properties, again, the heads being hydrophilic, being polar, being water-loving, and the tails being hydrophobic, non-polar, water-fearing, you create this selective boundary where polar molecules are not going to be able to cross the membrane because polar molecules and non-polar molecules don't interact. So molecules like glucose, a polar substance, is not going to be able to cross the membrane freely because it means it has to interact with the hydrophobic tails of the phospholipids, the internal part of the membrane. But non-polar molecules, they're going to be able to move across the membrane pretty easily because they're going to be able to interact with those hydrophobic tails. Now, of course, that might be bringing up a question in your mind. Well, wait a minute. How could a non-polar molecule like oxygen or carbon dioxide make its way across the hydrophilic part, right, the polar heads of those phospholipids? And that brings us to the next piece of the membrane puzzle. That while the chemical properties of the phospholipids are ultimately going to determine what gets in and what gets out, when it comes to polar molecules, we have to build out our understanding of how the phospholipids themselves are interacting with each other to appreciate how molecules that are nonpolar are actually getting across the membrane. So I'm going to skip these for now. These are great practice questions for later. So the first piece of the puzzle to help us understand what's happening is that, and you saw this already in the video that you watched today and in the Edpuzzle video that you watched last night, that the membrane is not solely composed of phospholipids. It's composed of other molecules as well. Different types of proteins. You were introduced to integral and peripheral proteins, as well as molecules like cholesterol that are found within the internal nonpolar portion of the membrane. But I think what's super important as we think about how it might be possible for hydrophobic molecules to make their way past the hydrophilic polar heads of phospholipids is to actually look at how all of these molecules, the phospholipids and the proteins alike, are interacting with each other. So my hope is to come up here. Of course, now it wants to move slower than death. All right, can you see this graphic on your screen okay? All right, so my hope is you're seeing some of the phospholipids, right? You can see those heads and those tails. They're organized into a bilayer, okay? And then these long tubular structures, right? The longest one kind of in the middle right now, that would be an example of your integral protein that you were introduced to in the Edpuzzle video last night. And these smaller proteins, they would be examples of the peripheral proteins that are finding themselves embedded into one, but not both layers of the membrane. So as I press play, what I want you to begin to appreciate is that these molecules that make up the membrane, they're not static, right? They're constantly moving. They're bumping into each other. They're moving past one another. Sometimes they even flip from one layer into the next. Now that's rare, but it can happen. And I'm showing you this, and I'm going to keep showing you this for a couple of rounds, because what I'm hoping you're noticing, once I can pause it at just the right moment here, is that every now and then, I got my pen up, gaps form between neighboring phospholipid molecules. So those heads are still hydrophilic. They're still polar structures, right? They're going to prevent, normally, nonpolar molecules from being able to cross the membrane. Because again, nonpolar hydrophobic molecules, they're not going to interact with hydrophilic polar parts of molecules. Like does not dissolve unlike, right? Like dissolves like. But it's because these molecules are in constant motion, right, that sometimes these gaps will spontaneously form between phospholipids, and that's going to provide the space for nonpolar molecules to zip through the membrane. Because once they get through that gap, right, they're going to find themselves in a hydrophobic nonpolar environment that's made up by the tails of the phospholipids, so like dissolves like there. And then as long as there's another gap, right, that forms spontaneously on the other side of the membrane, the nonpolar molecule is going to be able to find its way out, and in this case, out of the membrane into the next part of the cell. So I want to pause there because there's a lot going on. I can press play again. Does that bring up any questions for you? So we looked at the composition of the membrane, right? We've looked at how the composition of the membrane ultimately determines what gets in and what gets out of the cell. What we're adding on now in these last few minutes of class is that the molecules that make up the membrane are in constant motion, right? That the membrane is a fluid. And that's really important to us because we can use it to explain how molecules that we might not expect to be able to cross the membrane actually can make their way across the membrane. All right. So there's no questions. Just about done here. I want to leave you with one last example of why all of this matters. And if I have a minute at the end here, I'll even tell you a quick story from my past in teaching that I share every year when we talk about this content. All right. Pop up for me. There it is. So because the membrane is fluid, because those molecules are in constant motion, we can do things like this. So what you're looking at on the screen right now, on the left-hand side, this is a human egg cell. On the right-hand side, you have a very, very narrow glass pipette. And if you look inside, let's see if I can highlight it for you, right here. What do you think might be inside that glass pipette? If what's on the left is a human egg cell.
Maybe water.
So there is certainly going to be water inside the pipette, because everything that you're looking at right now is happening under the microscope. So the egg cell, the pipette, and what's inside the pipette, they're all in solution for sure. What if I were to press play here? Maybe that will help you to answer the question. Maybe you can even see that thing that was inside the pipette moving. I can play it again, of course. What you're seeing here is a sperm cell being injected directly into an egg cell. It has a name. It's called intracytoplasmic sperm injection. And the reason why I'm showing you this is because, as you watch the video, you can actually see, right, that sperm cell inside the pipette be injected directly into the egg cell. But it's this part right here that's really important, because none of this would be possible if the membrane was a rigid, solid structure, if it was non-fluid. You can see the membrane bends and flexes as that pipette is pushed against it and eventually inserted into the cell itself, so that the sperm cell can be injected into the cell directly. And in fact, at the very end, I think you can see it, the pipette is pulled out, and the egg cell doesn't rupture or burst like a balloon would if you poked a balloon with a pin, right? All those phospholipids that make up this fluid membrane that kind of separated and parted as the pipette was inserted into the membrane, when the pipette was pulled out, all those molecules moved together and sealed the gap. So the membrane and all of the molecules that make it up are fluid in nature. They're constantly moving, and because they're moving, we can do things with cells that allow for processes to take place that otherwise would be impossible if the membrane was a rigid structure. So of course, when you look at intracytoplasmic sperm injection, what you're really looking at is a fertility treatment. There are human beings, for reasons that are no fault of their own, that cannot conceive children because either the egg cell cannot be fertilized by a sperm cell directly, or the sperm cell can't fertilize the egg cell directly. So biologists have come up with a way to kind of circumvent that process and inject the sperm cell directly into the egg cell. And the quick story that I wanted to share with you is, gosh, this is my 18th year teaching. So in one of my first years teaching back at the other school that I taught at, there was a student in the class, I was showing a video similar to this one, and she didn't stand up, but she raised her hand and said, "Mr. Bergeron, how dare you show us this?" I said, "What? How dare I show you this? I'm just trying to show you an example of the fluid nature of the membrane." "Well, but Mr. Bergeron, you shouldn't be showing us this because this is doctors and scientists playing God. That this process is not normal. It's not natural. Nobody should be able to do this." And I said, I'm sorry, I didn't mean to offend you. I'm just trying to show you an example of why the molecules that make up this membrane, because they're existing as a fluid, right, that the membrane can bend and flex and can accommodate this pipette, and things can be injected into the cell. And you can pull the pipette out and the membrane seals back on itself so that the cell doesn't die, it doesn't rupture, it doesn't burst. And I guess that kind of satisfied her at the moment, until the next day when she came back to class. She said, "Mr. Bergeron, I have to apologize for what I said yesterday. I went home last night and I was telling my mom and dad what I learned about in class and how offended I was, and my reaction to what you were sharing. And then they told me that I was the product of that very process that I found so offensive. She was a test tube baby. She was the product of in vitro fertilization, of intracytoplasmic sperm injection. So be careful what you object to, because sometimes what you object to is actually responsible for how you came into existence. All right. Well, before you go, if you could, in just a minute or less, if you have to go, it's totally understandable. Is there something about what you experienced today that you might bring home and tell somebody else about what you learned? Can you summarize that in a sentence or two before you go? And as soon as you have added something, you're welcome to sign off. And gosh, I guess I'll see you again on Monday.
Thank you.
Thank you.
Here they are in the chat.
Have a good day, Mr. Bergeron.
Isaac.
Okay.
Thank
you.
Bye, Mr. Bergeron.
Isaac.